I hope I'm not too much interrupting.
No, that's the reason that I didn't want to do it in the lab, because everybody has to stop working when they're filming, and I can't afford it.
Yeah.
And they complain, of course.
Why is there so much interest in interviewing?
Well, because there is being used so widely in the U.S. and so many states have approved it, and the same is happening in Europe. So when something which is not well known is being so widely used, people come out and start asking questions: how, where, what are the type of diseases it can do, what is the scientific background, what are the side effects, and so on and so forth.
Did you ever expect that when you were starting out that cannabis and marijuana will become your life's work?
Well, this is one of the topics we've worked on. I am a natural products chemist. I work on the chemistry of natural products. On the chemistry, plants produce a lot of compounds. Contrary to humans, so mammalians, all of us produce the same type of compounds. We produce the same type of compounds, more or less, like a cow or a duck or a pig. Not with the plants, because we have an immune system. Plants don't have a specific immune system. Their immune system is based to a large extent on compound they produce, and every plant family has more or less the same type of compounds. But there are many families, and therefore there are thousands of compounds in the plants. And most of these compounds, their structures are unknown or their activities unknown. So my group has worked for the last 50 years on the chemistry and biology of natural products. You see, most people are interested in the plant. Cannabidiol, THC in the plant, and so on. So we work on the plant. Identification of the active compound. See, the active compound, THC, was only partially known when we started work on it. The structure was unknown. It was not available for research, and of course, that's a major problem. This is contrary to morphine, which had been isolated from opium 150 years previously, or cocaine, which had been isolated from coca leaves 100 years previously. And of course, they had been investigated thoroughly. Here we had a strange situation. There is a plant that's being widely used in various forms: marijuana, hashish, etc., etc. But the science behind it was rather weak, because the active compound or compounds had never been isolated in pure form, and they could not be investigated. So in the 60s, we went first into that direction, isolating the THC and many other compounds. And we re-isolated cannabidiol and quite a lot of other compounds, quite many of them new compounds that had never been isolated previously. We identified their structures, and then we started looking at the metabolism, the activity, and so on. And this went for quite a long time. This is essentially the first stage of cannabinoid research: work on the plant, chemistry, biology, clinical effects, and we did some clinical trials just as well. Now, this is something that we did in the 60s, early 70s. We did some clinical work, and I can tell you about it later. But that was the first stage. One thing that was not known at that point: how does it work? What's the mechanism of cannabinoid work? It turned out that there are receptors, proteins in the brain and in the body, that THC binds to them. And these receptors, there are two of them. One, when activated, causes the well-known effects of marijuana, the high. The other one does not. Binding of THC to that particular receptor causes only protective actions, and doesn't cause the high. So THC binds to both these receptors. Now, we don't have receptors in our body because there is a plant out there. We have receptors in our body because we produce compounds that activate these receptors when needed. So we went ahead looking for these compounds, and we found eventually 2 compounds that our body produces. Chemically, they're completely different from the compound that the plant produces, but they have the same activity, more or less. So we called one of them anandamide, and the other one 2-AG. Ananda in Sanskrit, of course you know Sanskrit.
I speak it every day.
Yes. So we found that Ananda is supreme joy. Anyway, we called the compound anandamide, and we elucidated its structure and synthesized it, and the same with the second compound, 2-AG. And these 2 compounds are present in the body, made by the body when and where needed. And they act when and where needed. For example, if somebody has been injured and there is pain, the body will produce anandamide and 2-AG in that area in order to lower the pain. But they are involved in a huge number of additional actions in the brain. As a matter of fact, researchers at NIH, National Institute of Health, in the US had a review, and they said these 2 compounds, as a matter of fact, the whole system of endocannabinoid system, the receptors, the compounds, and so on, are involved in essentially all human diseases. This is a very strong statement, but they are because they do several things. They regulate some of the other neurotransmitters in the body, and they have actions of their own. So they are extremely important, and therefore there has been a huge amount of work on them. Thousands of papers have been published on anandamide and 2-AG, and we know quite a lot about their activity within the body. So we see that as the second stage of cannabinoid research, showing how this endogenous cannabinoid system works in the mammalian body, in our body, and how it is associated with everyday life, mood, happiness, sorrow, and so on, and with disease states.
Is it sort of like like a part of the immune system somehow?
Well, it is parallel to the immune system. The immune system guards us against all kinds of things happening to us. Without the immune system, we'll be dead within 24 hours. Basically, activation of the endocannabinoid system of the CB2 receptor is apparently extremely important in guarding us against all kinds of things that are happening in the body, and we want to prevent them. We don't know enough about it, but we know quite a bit, and we have seen that in many diseases, activation of CB2 is important. It does not completely parallel the immune system, maybe it adds to the immune system. And at some point we shall know, I hope much better, how these 2 systems work together.
For someone who gets interviewed twice a week about this, you seem very, very energetic.
Well, I have to be at my age, but but now we have moved to the 3rd stage of the cannabinoid research, and there are very few people interested in that stage at the moment. I can tell you in a few words what's going on. The endogenous cannabinoids we have in the body are formed are not very complicated compounds. They are formed from fatty acids, like oleic acid, which is present in olive oil, and amino acids, which form the proteins in the body. So here we have a simple, relatively simple compound. But the body has prepared almost 100 of these, maybe more, of compounds of the same type, fatty acids bound to amino acids. And it doesn't make 100 compounds because it has nothing to do. It makes them because of it knows how to make them, and it needs them. And we have found that these compounds, about 100 of them, those that we have investigated, are of major importance in our body. So this is the 3rd stage of cannabinoid research. We found that one of the compounds acts in osteoporosis, the breaking down of bones. The background of that, women after the age of 50, with all the changes that are happening in the hormone levels, then they should have a lot of bone damage, osteoporosis. Not all women have that. And the reason is women, as well as men, but women in this case, produce a compound which is closely related to the endocannabinoids, closely related to anandamide, which blocks osteoporosis. In another field, with Professor Shohami, we found that compounds of this type affect brain damage. And if somebody has brain damage, the brain starts producing them and lowers the brain damage.
Wait, but if someone with osteoporosis creates these compounds, then how come they still have osteoporosis?
Well, they do not block completely. They act against osteoporosis. The body starts breaking down the bone. See, our bones are not stable in the sense that the same bones that you had when you were 6 years old will be when you're 60 years old. The body changes them. I mean, breaks them down and builds them again. Breaks them down and builds them again. There are cells that build the bones and cells that break the bones. So these compounds, endogenous compounds, limit the action of the cells that break the bones and enhance the action of the cells that build the bones. So it's a regulator in this case. The same is true for brain damage. We recently found something else which is also of major importance, I believe. We have to find out. Our body makes one compound of that type that tries to block certain addictions. We so far identified one compound and published it, showing that it blocks nicotine addiction and it can block the withdrawal effects, both of nicotine and of opiates in animals. We haven't tested that in humans. That's complete. That's the next phase. So if the body does that, it's a major advance towards using such compounds as anti-addictive compounds. So here we are at the third stage of cannabinoid research, working on compounds that are somewhat related to the endogenous cannabinoids, but have a huge effects in many other medical situations. I believe that within the next decade or so, we shall have quite a few cannabinoid-based drugs on the market in various fields. Let me give you a few examples.
Yeah.
We work on cannabidiol, a major compound found in cannabis, which is not psychoactive, it doesn't cause the high. And people would have thought, okay, so it's not of major interest. It is of major interest because cannabidiol is a major possible drug. We have seen that in epilepsy, it blocks epileptic attacks. We published that 35 years ago. Cannabidiol blocked epileptic attacks in patients that nothing else was helping them. What happened? Nothing for 30 years. Just the neurologists were not very interested. Nobody was very interested until parents found out that plants that contain a lot of cannabidiol, maybe they saw our work or maybe they just learned it from other sources, they found that cannabidiol helps children with epilepsy. There are children that have 20, 30 attacks a day. Well, cannabidiol helps them. So our work on the prevention of epileptic attacks by cannabidiol is an important medical step forward. Now, about 150 children in Israel are getting extracts with a huge amount of cannabidiol that blocks their epileptic attacks. So these children would not normally will not be able to develop with 20 attacks a day. Now they are more or less normal. In the US, a major study was done based on what we did 35 years ago. Yes, they showed that it's of major importance in epilepsy. So here we have a medical advance based on the basic science on identifying cannabidiol, looking at cannabidiol in animals, finding out that it's anti-epileptic, then going into humans. It doesn't always work that way. We showed that in diabetes type 1, which is an autoimmune disease, cannabidiol is of major help. Well, nobody has done that in the humans, which is a shame, because it helps in animals, exactly the same mechanism. It helps with diabetes type 1. Why it has not been tested in humans, it's beyond me. It costs a lot of money to do clinical trials. But I have no doubt that when tested in humans, it will show the same effect. Cannabidiol has been shown to be an excellent anti-schizophrenic drug in schizophrenic patients. Now, schizophrenia is not a minor disease. 1% of the population in every country has schizophrenia. So we're dealing with millions. And there are drugs against schizophrenia, but they cause side effects. Cannabidiol does not cause side effects. And once it is pushed forward, chances are that it will become a major anti-schizophrenic drugs. So we see a lot about autoimmune diseases, namely diseases in which the body attacks itself. Why the body attacks itself is a complicated question. We, in most cases, don't know much about it. Cannabidiol has been found to act very positively in autoimmune diseases. As a matter of fact, a group here at the Baileyton Hospital looked at something that is more or less like an autoimmune disease. In many cancer patients, one has to exchange part of the body, the bone marrow, with bone marrow from outside. But the body doesn't like bone marrow from the outside, and it attacks the bone marrow, the new bone marrow, and the new bone marrow attacks the body, and the patient is sick. It has to be done because there is nothing else that can be done with these cancer patients. But many of them are sick, and this is called graft versus host disease, which is an autoimmune disease more or less. Well, a group at Baileyton thought, I was a little bit involved with them, a group at Baileyton Hospital thought that maybe, as cannabidiol is an autoimmune disease drug, let's give them cannabidiol. They gave them cannabidiol, and the results were outstanding. It definitely helps in graft versus host disease. So a company has taken up now this particular information by the Baileyton Hospital, and they are developing cannabidiol as a drug in the case of graft versus host disease.
It's really interesting that we think of cannabis medicine as cannabinoid based, but we don't ever think about it as endocannabinoid based.
It will be 10 years from now.
Should it be?
Of course.
Why?
Because these are compounds that we make. If our body makes a compound, it makes it for a certain reason. And we more or less know what the reasons are. And there is no doubt that at some point people will start using it. Let me give you an example from a different field. There is a compound in our body called dopamine. And there is a major dopaminergic system. Well, it was known, but it was slowly discovered that the lack of dopamine causes one disease, and too much dopamine causes another disease. Too much causes schizophrenia. So in order to block, if there isn't enough, in Parkinson's disease, for example, one gives dopaminergic type compound to the body. If there is schizophrenia, one tries to lower that. So the body has to have a certain amount of dopamine, not too much and not too little. The same is probably true for anandamide. People have come out and claiming, without any major proof that migraine, for example, is due to maybe lack of anandamide. Maybe. But of course, it's very difficult to do work on migraine, because if you ask a mouse, do you have migraine? It doesn't want to answer you. But basically, it seems to be, maybe it's true, so it has to be investigated. If in many of these diseases we find that anandamide or 2-AG, the two endogenous cannabinoids, there is too much of them or too little of them, maybe we can do something about it.
What, so can endocannabinoid-based diseases that are related to the endocannabinoid system be thought of as a nourishment issue? Like, in other words, that if we're not having enough, not making enough endocannabinoids, we need cannabinoid nourishment for some reason?
Well, possibly. I mean, this is still being investigated. But the fact that the endocannabinoid system is involved in a huge number of diseases, then obviously we have to think along those lines.
I've heard very interesting things about clinical trials. Some doctors have told me and researchers told me that they think clinical trials might need to be adjusted or even reinvented, rethought of, because for cannabis-based medicine, because its effect seems to be so individual. Dedi Meili and someone else said they completely disagree with that it has to stay exactly the same. I think Moshe Hod also did say exactly the same, and that they have to do just isolates, and build up a pharmacology of which cannabinoids work together for what. What do you think about that?
Well, you have good research and you have bad research. If you don't do it the right way, it's bad research, and the results are not worthwhile. But today, advancing in the chemical medical side has to be done by good research of originally in animals and then in humans. The clinical trials have to be done according to rules that are well accepted by clinicians. Now, if somebody does a lousy research, well, it's a lousy research. But the good research is good research. One has to look at the research as it is done. Without clinical trials, today a compound cannot become a drug.
But for cannabis medicine, can we do clinical trials the exact same way we do it for other medicines? Same methods?
Yes, one can take a compound, cannabidiol, and give it to schizophrenics. Take the same solution that contains the cannabidiol, but does not contain the cannabidiol itself, only the solvents and all the formulation there, and give it to other patients as the placebo. These are basic rules, and for example, the German group that published on cannabidiol in schizophrenia did an excellent job. So the work that was just completed in the US on epilepsy, a large number of epileptic children, it was an outstanding research proposal. It had to be approved by many other clinicians. It costs, unfortunately, millions to do it, but once it's well done, it should be well accepted. Clinicians have to base their work, their day-to-day work, on clinical trials. And these clinical trials have to be outstanding.
Can you do clinical trials with stuff that's not just isolated compounds, like with full spectrum plants?
Well, it is more difficult because it's doing it with a single compound. One can say what are the amounts of the single compounds, how often, how it has to be given, and so on. When a clinical trial is done with an extract, then one has a problem because the extracts can differ. So I assume that the people you talked to and were negative thought about extracts and not about well-defined compounds. This is true. Working with extracts may be very complicated, And this is because of something that we discovered many years ago, which is called the entourage effect. The entourage effect means that there are compounds in cannabis that modify the activity of THC. They by themselves have no activity, but they modify the activity of THC. So if you have different compounds every time you do a clinical trial, or an animal trial as a matter of fact, with an extract which is not exactly the extract you had previously or somebody else had previously, then obviously you cannot compare because of the entourage effect.
So that's so is that a barrier for research? How do you overcome that?
Well, you work with well-defined material.
So about isolate and full spectrum, there are people who have told me very, very confidently and adamantly that isolates are not helpful. But I've definitely heard the opposite. I've heard isolates can be very helpful. Some people are saying only full spectrum is helpful, and you have to have THC to make the CBD become, useful. What do you think about that?
These are very general statements. You have to go into very specific cases. Pure cannabidiol is outstanding in many diseases. There are a lot of people that claim that one has to use extracts in order to get better effects. Maybe in some cases this is correct, but in order to do a clinical trial, one has to do it with well-defined extracts. So this is where we stand.
Do you think so? Is pure cannabidiol useful in many indications that you've seen so far?
Yes, as I said, we did work on epilepsy with 100% cannabidiol, pure, crystalline. The people in Germany did work on schizophrenia with 100% pure cannabidiol. The work in epilepsy in the States, the clinical trial, was done with compound which was almost pure CBD extracted from the plant. And the reason they did it is because an extract from a plant falls under different legal definition in the States. A pure compound has, that's what I understand, I'm not a legal person, a compound that is pure falls under jurisdiction of the FDA. Extract of a plant is something else, so it's easier to do. We have made derivatives of cannabidiol which are better than cannabidiol itself. We have published, for example, cannabidiol with a fluorine atom on the cannabidiol molecule. So it's a fluorocannabidiol. We have published that in collaboration with a group in South America. We have shown that it is pretty good in anxiety, lowering anxiety, pretty good in acting on other psychiatric diseases. We have shown that cannabidiol itself is pretty good in these things. We have recently shown that another derivative, which is a derivative of the acid of cannabidiol, helps with depression. So chances are that at some point some of these compounds will become major drugs. You see, depression and anxiety, for example, are major diseases in Western society. Huge percentages of the population have anxiety or depression on and off. And we need compounds that do not cause side effects. We need simple compounds, or compounds that are, one doesn't have to be anxious taking the compounds against anxiety.
What do we know about cannabis in pregnancy and in birth and in women's health in general? Is that and its effect on the mother and the fetus?
Well, most women that still take cannabis say that nothing major has happened. For some time it was thought that the children that are born may be smaller and develop more or less normally. But basically, I would strongly recommend not to use anything during pregnancy, not cannabis or not any drug if possible, because it's extremely difficult uh, to know exactly what's going on. If you have a drug which causes damage to the fetus in 50% of the cases, this will be seen immediately. But what happens if the damage is seen only in one fetus out of 1,000? It is not seen in any clinical trial, but the one damage is one damage. That particular damaged baby is damaged. And we don't know. It's extremely difficult to prove anything of that sort. I mean, we need statistical proofs. It is very difficult to work with effects that, with things that affect, I don't know, 0.5% of the population or 0.5% of the fetuses. And what I would normally suggest, if a pregnant woman asks me, may I take cannabis because it lowers the vomiting I have during the pregnancy, I say, well, too bad. Just suffer the vomiting and don't take anything, any drug.
But isn't that true for any disease that anyone has? Because you can say, well, we haven't checked it on 10,000 people yet, so maybe it will do damage, even though it helps many people.
Well, this is probably true for all kinds of minor effects, but there we know that in a disease it helps the disease. Pregnancy is not a disease. Pregnancy is a normal thing.
So are there any things that you would have done just since 1964 was the THC isolation, right? So since then, is there anything you would have done differently that you think would have changed in any way the way that it's the industry, I'm calling it, has gone?
Well, scientifically, probably not. Probably I could have got more money to do more research, but basically we went the right way. What I should have done better is try to get industrial interest, and still, although we know that these compounds have activity, cannabidiol, derivatives cannabidiol, they are still not sold as defined drugs. And possibly I could have pushed more for that. For example, Teva, the biggest company in this in Israel, the biggest pharmaceutical company, is not really interested, was not interested. Chances are that we could have pushed more for that, but we didn't.
Yeah. So okay, so where do you see this going in terms of cannabinoid medicine getting into pharmaceutical drugs? How do you see that process happening?
Well, things are changing. Israel, as a matter of fact, is pretty well ahead of many other countries. Medical cannabis can be administered here. One needs an approval by the ministry or by physicians that have that are associated with the ministry. There are about 35,000 patients that have approval, which is a huge number for well-defined diseases, particularly in diseases that are not associated with psychiatry, almost everything else. And today there are, I don't know, probably 50, 60 labs in this country that do research in cannabis and various aspects of cannabis. The clinical trials today, contrary to what was in the past, cost a lot of money. I'll give you an example. When 100 years ago, insulin was discovered, and insulin is the compound that our body makes in order to regulate the sugars in our body. It was used immediately as a drug. It's an excellent drug, still being used in one way or another, the insulin. Anandamide, which we discovered 25 years ago, has never been given to a human, never. Insulin was given within 6 months. Anandamide probably is a pretty good drug, but it has never been given to a human because a company has to be interested and has to spend many millions of dollars to develop. So,
are you surprised when you look around and see just how big this is becoming, on how accepted this is becoming, and how much research there is relative to when you were just?
well, I'm delighted. I'm delighted that the medical part of it is developing slowly, but it is developing. One should separate the medical use from the recreational use. The medical use has to go according to well-established medical routes. It has to go through toxicity and animals and then into the humans. It has, in order to be accepted by the physicians, it has to follow the well-established medical rules. Recreational use is a social issue. Recreational use has to be decided by the population. Whether it's toxic or not, that's something else. Obviously, populations all over the world have decided 100 to 200 years ago that smoking tobacco is fine. The fact that people die out of smoking tobacco doesn't matter. It has been socially accepted. The same is true for, let's say, whiskey, 50% alcohol. Definitely not very healthy, but it is approved. But this is a social issue. One cannot come over and say we should be allowed to use cannabis recreationally because it helps with epilepsy. What's the logic there? There is no logic. These two things should be completely separated. One is a medical issue, the other is a social issue.